Is the recording started or not? Yeah. Okay. I'm sharing the slides with you. Is it visible to you?
Yes, ma'am.
Yes, ma'am.
Okay. So today we'll be starting with a very important chapter of molecular biology, the DNA replication. Are you getting some noise? Is it creating problems? The sweeper people
No,
are
not that much.
cleaning. Okay, no
Not that
problem.
much.
No problem for you. Then it's good. Okay. So we were discussing about DNA replications. It's a very important chapter in molecular biology. I'm not putting in slideshow mode because I have to show you some, to draw some things. Okay. So this is Is this visible? DNA replication?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Okay. So what is actually happening when a cell is dividing into two? So the genetic material, the DNA also becomes two, and to each of the cell, daughter cells, the DNA then passes. Okay. So how this phenomenon occurs, that is very important, because if the DNA, first of all, the DNA replicates into two parts, and DNA replication is very interesting in the sense there are certain salient features in DNA replication which you need to learn. So all of you know how the DNA looks like. It's a double-ended helical structure, isn't it? Few features of the DNA helical structures that we have discussed in biochemistry. Can anyone just recall and tell me at least 2-3 points? DNA, what are the features of the DNA double helical structure? What are the salient features?
Ma'am, the strands are anti-parallel.
Anti-parallel.
One is 5'3", one is 3'5".
Yes, and that is because of what? Why this 5-3' means?
Pentose shortkora. Pentose.
Phosphorus. There were the phosphorus which are at the
Yes.
3' end, and the because of the number of the way, the direction of the phosphorus. One is in the upper hand, another is in the lower hand. So these two strands are anti-parallel in nature. Okay, the first thing. Now anything else you want to add?
Ma'am, the nitrogen base, purine base and
Base
pyridine.
pairs are added.
Their bases are always there, purines and pyridines, but the main
Hydrogen
important
bond.
thing is the base pairing. Okay, so the purines and the pyrimidine interact in the such a way that A pairs with T and G pairs with C. This base pairing is based on the Chargaff's rule, and it is very important. This thing is being followed in the DNA double helical structure. AT, 2 hydrogen bonds are there, and GC, there are 3 hydrogen bonds between the two molecules, right? So these are two very important features. Anything else in DNA double helical structure? They're anti-parallel, they're complementary in the sense that they are base pairing, they are followed, and also they are polar in nature. Okay, so you all know about this. So what is happening in DNA replications? DNA replication doesn't occur in any other place, not on every place. There are certain points where the DNA starts replicating. Okay, and that particular point is known as the origin of replication. Right. So first point, note it down. It's a DNA replication. It starts at the origin of replications. So at that origin of replication, DNA actually forms a like fork-like structure, replication fork, which is known as a fork-like structure. It's open up, and what is happening over here? It is very interesting that the two strands of DNA just they separate. They separate with each other, and each of the two strands. I'm showing in my laser part. Again, I have to go back. Okay, so these are the two strands. Let's say I just the double helical structure is now shown in a ladder-like form. AT, and GC base pairing is shown here. So the two strands are complementary to each other, and they are forming the base pair rules. If for DNA replication, the two strands separate with each other, and each of this strand as well as this strand, both act as two templates. Templates, you got it? No, templates means the set on this, based on this strand, the new strand will be formed. So that is why it is known as the template. Okay. So this on the first strand, one strand will be formed, on the second strand, another strand will be formed, and at the end, we are getting two copies. So if you look into this color code, these two strands, first of all, the DNA has two blue color. The two strands are colored in blue, but when we are forming these two new strands, this is the newly formed, which is shown in magenta or pink. There is another new strand that is formed in pink, and at the end we are getting two different strands. So if you look this DNA, the parent DNA, which was both the strands were in completely blue color. Now what we are getting the end product, there are two DNAs in which one of the strand is blue in color and another is in pink, and another strand also again blue in color and pink in color. So if this was my input for starting point, and this was my output, the results. So if you look into this, what actually do you get? So the DNA, the two strands were same, like in a being blue in color, but right at the end we are getting two daughters DNA, which are actually having one blue strand and one is pink strand. And it's the same case with the other daughter strand. So if this is my parent DNA and this is my daughter DNA, what I can say is that this newly formed daughter DNAs are actually getting one strand from their parent and one strand is newly synthesized. Are you getting clear, class? This is the fundamental thing in DNA replication, that this new daughters that are formed from the parent are actually having one from the parent and one they are newly synthesized. And that is the reason DNA replication is known as semi-conservative. Semi-conservative DNA replication. That is a very, very vital question people often ask. Why DNA replication is semi-conservative in nature? If it had been conservative, what would have been formed?
Only one strand.
Yeah, this pink strand would not have been, it's completely blue. It should have been completely blue if it would have been completely conservative in nature. Okay, so it is not completely conservative, it is semi-conservative in the nature, in the sense that the newly formed DNA has one strand from the parent and one is completely new. Clear? What is DNA semi-conservative replication? This is the main fundamental thing, and how this procedure takes place, we'll discuss about it.
Ma'am, please repeat the template.
Template. Okay. So first of all, this is my parent DNA. I have got this DNA, and from this DNA, it is actually forming two copies. Okay, this parent DNA is forming two daughter DNAs. So what exactly is happening over here? The DNA is a double helical structures coiled with each other. What is happening first? These two strands separate first of all, and then what is happening? And newly strands are formed based on this template regions, based on the parent strands. So what exactly is happening here? It is they are following the base pairing rules. So when they are opening up, here G is there. So new nucleotide C will attach to this. Then T will be having A. Then A will attach to T. And then C will attach to G. Follow the pointer, whatever I am showing. Similarly, in this strand, the other strand, C will pair with G. A will pair with T. T will pair with A. And G will pair with C. But these, the shown in pink color, are the newly ones. And at the end, we are getting two copies of the DNA. And in these two DNAs, one strand, this is the first strand, this is the parental one, which is complete. It looks like in the same thing as the first one, the blue in color. But the second strand that is newly synthesized is pink in color, color coded one. This is just schematic diagram I'm showing you. Okay. So I hope you are getting my point. What is semi-conservative DNA replication? Any doubts, class? So DNA replication is very important in the sense that it follows the semi-conservative replications. The newly synthesized daughter strands, daughter DNAs, these are the two DNA daughter strands, and one of them are receiving one strand from the parent and one is newly synthesized. This is known as DNA semi-conservative replications. Now, how scientists came to know this DNA replication is semi-conservative in nature. So, very clear that they performed certain experiments and then only this was proved, isn't it? So Watson and Crick actually gave the DNA structures, as you all know. So they actually hypothesized this semi-conservative replications after the publishing of the structure of the DNA. And this method, semi-conservative replication, was actually proved by a very famous experiment done by Matthew Meselson and Franklin Stahl. I advise all of you to learn about this Meselson and Stahl experiment. Just search in the internet, or you can go to the CBSE books also. They have very nicely explained how this has happened. DNA is semi-conservative. They have used actually the radioactive nitrogen to show that how this DNA replication is semi-conservative in nature. But basically, if someone asks you what is semi-conservative replication, that you must say that to the point that the newly synthesized DNA molecules, the daughter DNAs, are actually having one strand from the parent and one is newly synthesized, and that is why it is known as the semi-conservative in nature. Okay, semi-conservative. But again, based on your marks, if this is 5 marks question or 2 marks questions, you have to write the proper mechanism, how it happens. And how it happens, we're just going to discuss right now. DNA replication. But basic idea समझ में आया? What is DNA replication? How it is semi-conservative in nature? Class?
Yes, ma'am.
Okay, good. So this is something that happens. So as I said, that DNA replication starts at certain point. That point is known as the origin of replication. At the origin of replications, the DNA actually forms a certain opening. You know, DNA is a very, very big molecule, so entire strand cannot just open in the same entirely from end to end, separate cannot. This is because it will take a lots of energy. And that is why there's a replication occurs at certain point. That's the original replication, and at some opening it's done. And that opening is known as the replication fork. This is the replicating fork. This is like a fork, like it's a fork. Now look how this is, like it's open up. So this Y-shaped junction, if you look like this is a inverted Y, isn't it? This looks like an inverted Y. This Y-shaped junction is known as a replication fork. Now going back, the blue and red color, if you look, so this strand 5' 3' and this 3' to 5' anti-parallel to each other and complementary in nature, so they open up. Now newly strands DNA, how it forms, it takes the help of certain enzymes. Okay, for DNA replication several enzymes are involved, but for your course purpose, try to remember at least 2 or 3 enzymes. 2 to minimum 2 enzymes at least you remember. One of the important enzyme that is required for DNA replication is the DNA polymerase. DNA polymerase actually starts the the replication thing. You can see here what is actually happening over here. I need to draw a picture so that you can understand this thing. Sorry, class. Let's see. Okay, so we have our DNA. This is not opening.
Ma'am, one thing. Ma'am,
Yeah.
one part is continuous and one part goes discontinuous.
I have not yet discussed that mechanism. Is not yet started.
Okay, ma'am.
Discussing.
Ma'am, can you just show the last slide, ma'am, for once?
Yeah, sure. See.
Can you please slide show it for once?
Okay. Okay.
Thank you, ma'am.
Fine. Okay. So, as I said, DNA is a double helical molecule. It looks like this. So my pen is having some issues. So it's okay. कुछ हुआ है नहीं लग रहा है. Anyways. So this is how a DNA. I'm very bad in this eraser. So this like something DNA. ठीक है? DNA की तरह दिख रहा है, right? Class, can you see what I am actually drawing?
Yes, ma'am.
Okay.
Yes, ma'am.
Yes, ma'am.
Why it's not okay? So what is actually happening? This DNA is 5' to 3' and 3' to 5' that you know. Now for DNA replication, at certain point, as I said, the DNA opens up into like this. Let's say like this. Okay. So this strand, if you see, what is 5' to 3' and one is 3' to 5'. Let's say this is 5' to 3', another this 3' to 5', anti-parallel in nature and complementary to each other. What is happening here? Where the newly strand DNA are formed based on the template strands that we all discussed. What is happening here? Next step will be, I'll put another pen. So the DNA replication starts on the 5' to 3' direction. Means the newly strand DNA will form from the 5' to 3' end. So 5' to 3' end में जब हो रहा है, when it is going from the 5' to 3' end, that means that it is taking the 3' to 5' end as the template, because they are anti-complementary to each other. Okay, so this moves like this way, and this strand, this 5' to 3' strand, it is the leading strand, because here the there is the replication is continuous in nature, continuous in the strand, in the sense that, first of all, based on this base pairs, whatever base pairs are there here, A, T, G, C and all, so we are putting new nucleotides are coming up, and these are actually forming bonds with this already existing parents nucleotide. Suppose here I have G, then what will come? What is the base pair with G? C.
C.
Similarly, if it is T over here, then it will come, another nucleotide will come as
A.
A. And similarly, these bonds are being formed over here, and the new DNA strands is being formed like this way. And since it is forming from the 5' to 3' end, the template will be 3' to 5', the opposite one. Clear? So these, the nucleotides are actually being linked to each other with the help of an enzyme known as DNA polymerase. So the DNA polymerase is actually helping you to form the newly formed nucleotides are being added, and they are forming the new strands of the daughter strands, which is actually the leading strand, and it is continuous in nature. There is no breakdown over there. But in the other strand where this is happening the other way, here the strand, this is not completely continuous, it is a discontinuous where we are forming small fragments. These small fragments of DNA are known as the Okazaki fragment, based on the name of the scientist who actually discovered those, Okazaki, Japanese scientist. So these strands are actually small strands are formed, and this is actually happening in the other way direction, like this way. And at the end, these small fragments are actually joined together with the help of another enzyme. It's a DNA ligase. Ligase are enzymes we actually join, and at the end we are actually forming the lagging strand, because these are discontinuous. These are not forming in continuous manner. It is discontinuous in nature. Small fragments are formed, and they are joining with each other. So these are known as Okazaki fragments, and this strand is known as the lagging strand. So 5' to 3' is known as the leading one, leading strand, and the other strand, this is known as the lagging strand. Okay, so one of the leading strand is actually the continuous one, and the lagging strand is the discontinuous one. At the end, what we are getting, this replication. So this is the 5' prime, 3' prime, that is being formed 5' to 3', the parents, and then this we have the 3' to 5', and or the daughter strands are actually having this way, this way. So this is the 5' to 3', and similarly here 5' to 3', and then the base are being found within the different nucleotides, ATGC and all that. So what we are here getting actually, so initially I had this parent one which was incomplete, both the strands were blue in nature, then it is separating with each other, strands are separating, they are opening up and newly strands are formed. At the end we are forming two different daughter molecules which are actually having one copy from the parent, the blue strand, and the newly synthesized is shown in red. So the two strands, the two daughter DNAs are actually having one copy from their parent and one is newly synthesized, and that is why it is known as semi-DNA, semi-conservative in nature. And the main features of the salient features of the DNA replication is that, first of all, DNA replication starts at certain points known as the origin of replication. The DNA strands open up to form a fork-like structure known as the replication fork. The DNA replication starts at the 5' into the 3' prime end. So that is known as the leading strand, which is actually continuously replicating with the help of DNA polymerase, which is actually adding up different nucleotides, and that is nucleotides are following the base pairing rule, and it is based on the Chargraph's rule. A is pairing with T, G is pairing with C, and so on. So this strand is completely newly formed and it is leading in nature, continuous replication. The other strand of the daughter DNA that is being formed it is forming in discontinuous manner, in the sense that small fragments known as the Okazaki fragments are being produced, and these fragments are being joined through other enzymes, and at the end they are also actually forming the strand, and this are how we are getting two different daughter DNA molecules. So till now, any doubts, any confusion, class? What is leading strand? What is lagging strand? What is continuous replication? What is discontinuous? Origin of replication? Any doubts, class?
Ma'am, that strands from the parent DNA, it is referred as template.
Yes.
Okay, thank you.
Okay. That is template. Template in the sense that because of whatever the nucleotides are present at that strand, the newly synthesized DNA will actually form the complementary strand. Okay. तो आपको अगर पता ही नहीं होगा कि what is the things are there in, what are the ATGC sequence is there in the parent strand, how will you form the newly one? So because it is opening up, it is having ATGC sequence, then what will we join? The newly strand will have, as compared to A, it will form the T, G, then C, and so on. Okay. So two of the different enzymes. What are the two enzymes I actually discussed right now? If you remember, one of the important is DNA polymerase. Yes.
Ligase, DNA ligase.
DNA ligase or ligase, whatever you say, that is actually helping this Okazaki fragments to join together. Okay. So these two enzymes you can remember, and there are many other strands, many other enzymes which you can look into the books and you can find out. But suppose I am giving you the DNA strand. I am giving a question like this. Let's say, okay, this is again the same thing. How the picture is bipictorial. So this is the leading strand. Don't get confused if leading strand is written over here. Okay, leading strand means the newly strand that is formed. Leading strand is the 5' to 3' end. So ये वाला जो लिखा है ना, you don't get confused. It is written over here. It should be like here over. Okay, leading strand is the 5' to 3' directions, and this the template strand is 3' to 5'. The direction of movement is like in this way, 5' to 3' is continuously formed, and then Okazaki fragments are being formed, the lagging strand, and then they are joined through the ligase. So this one I had actually drawn in the for your reference from beforehand. This is a parent DNA 5' to 3' and 5' to 3' these are being formed. Small and then a daughter DNAs. There is a very nice tutorial on DNA replication. The YouTube link I can share with you in the MS Team itself. You can just have a look into that. Very nicely explained, you know. And suppose now I am giving you a task. Let's say. Now, if I ask you, I have. Yeah, I have a strand, let's say 5' to 3', okay, DNA molecule, and I'm giving you a sequence like this in exam. If it is given like T, then C, then G, and A, and so on, and you will be asked to write the complementary strand or the what will be the what will be the complementary strand. Then you will be able to write it, right?
Yes, ma'am.
So what will be the complementary strand?
Ma'am, it will be first we'll draw the line and it will be 3' to 5'.
Yes, that is very
And
important.
then
Many people forget. Don't forget to write the 3' to 5' direction. This is very, very important class. Now you have to write the complementary part. So if it is T, then it will be here.
A.
A. Yeah, and then it is C. If it is, then it will be
G.
C.
G.
अच्छा,
And then वो अच्छे से लिखा नहीं है.
Then
Yeah.
C. Then T.
So on. So ATGC, if you can remember, and don't forget to put these things. Okay, these are very, very important. If you do not put these things, then I'm going to deduct marks. I'm telling you, the class itself. Okay. So don't forget to put the complementary strands. The polarity is very important, and then you can put the things. So DNA replications till now, any doubts? So what are the salient features? Can just anyone of you tell me? Repeat the things that I discussed. What are the salient features of DNA replication? First of all, it is
semi-conservative.
Semi-conservative in replication. What is semi-conservative in nature means? Just one or two voices are telling. Girls, where are
like
you?
one part is from the parent.
And one is newly synthesized.
Newly synthesized. So these the students that people are answering, replying are the ones who had biology in their 12th. Those who did not had biology, are you facing any problem? Class?
No, ma'am.
समझ में आ रहा है? Okay. Are you
Yes,
following?
ma'am.
If you're not able to follow, then I am ready to discuss it because you know this is very, very important. I'm telling you again and again. And this is a very mark scoring question also, but people often make mistake. I have been teaching this from the past 3 years and I see that itna अच्छे से पढ़ाने के बाद भी and then students do mistake and you have to deduct marks. I don't want that my students should lose marks in at least DNA replication. That is very important.
Okay? और एक बार पढ़ा दीजिए.
और एक बार पढ़ा देंगे.
हाँ.
कहाँ पे डाउट हुआ है बताओ. पढ़ा तो हम लेंगे. कहाँ पे समझ नहीं आ रहा है. That is very important. जो पार्ट समझ में नहीं आ रहा we'll discuss again ना. So first of all, what is actually happening? Let me put on my camera there. Can you see me?
Yes, ma'am.
Okay. So
Yes.
I am taking let's say two wires. What is actually happening over here? So this is, let's say, my DNA. Okay. This is where what is happening. So it is actually turned like this helical structure, DNA helical structure. It is actually opening up like this.
Yes,
These are my two strands of the parent DNA. Can you see me, class?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
I don't know what is this. Has come up something. Okay. So suppose these are the two strands of the DNA. One is on the 5' to 3' and another is a 3' to 5'. अब मैं आपकी तरफ से पढ़ा रही हूँ, ठीक है? So this is from 3' to 5' and this is from 5' to 3'. Class. So my newly formed synthesis DNA will take place from the which end? 5' to 3.
Disconnect.
So 5' to 3' it will move like this. So it will take the other strand like the 3' to 5' end, and this way it will move from the newly strand. Actually, it is not like a train is moving like that. What is actually happening? Small molecules are being attached, small nucleotides coming up. They are actually forming base pairs with this parent DNA molecules, and they are actually joined through the DNA polymerase. So this strand is actually a leading strand, which is continuously being formed. So this continuous strand, and the other way, there are small fragments are being formed, which are known as the Okazaki fragments, which are actually then joined together with the help of another enzyme known as DNA ligase. Okay. At the end, what is happening? This and the new strand is formed one, and this another strand is being formed. So we are getting actually two DNA daughter strands, which is actually having one copy from the parent, one is newly synthesized. Clear? Till now again some issues, any problem class? अभी समझ में आ रहा है या?
Ma'am, can leading and the
Let's check
continuous
what
strand
is
once more?
एक बात एक. I mean, someone is telling. The girl is telling. Tell. Who is this?
Ma'am, the leading and the continuous strand, can you tell once more? That one I did not understand.
You did not understand. Okay, very good. ऐसे पूछोगे तब ना हम बताएंगे. Okay. So this figure, if you can see. Okay. So what is happening over here? See, look into this picture. This blue one is the parent DNA, which has actually opened up. This is known as a replication fork. So my newly synthesized DNA, this the pink one or the red one, whatever you say, it is synthesized from the 5' to 3' direction. And this is completely in a continuous manner it is actually forming. So one by one nucleotides are being actually added here based on the nucleotides that are present on the DNA, parent DNA. So newly nucleotides, like here if I have A, then G is coming, then T, then C is coming, and so on. So nucleotides coming up, and this molecule is actually synthesized in continuous manner. Okay, so DNA polymerase is actually forming bonds, and then this is a completely newly synthesized molecule. This is completely in a continuous manner it is being formed. So this is why it is known as the leading strand. It is actually leading and is moving from the 5' to 3' prime direction, continuously forming. So that is what was continuous replication. But on the other strand, if you look, these fragments are actually not completely in a continuous manner. These are not being formed. These are actually forming from the 5' to 3' prime direction, but in small fragment ways. And later these fragments are actually joined with the help of another enzyme known as the ligase or ligase, whatever you say. And that's why this strand is known as the lagging strand, whereas the DNA is discontinuous. The DNA formulation is discontinuous in nature. So one is leading strand, which is continuously being formed. One is lagging strand, where, yeah.
Ma'am, that is the doubt, that why it is discontinuous, why cannot be continuous, even if it is opposite in direction.
So that is what I am telling. No, that is because that is a phenomenon, and that is why there are special machinery for the DNA replication as well. So that is the thing that is been happening over here.
In the lagging strand, DNA polymerase is working or not?
Yeah, it's working. It's bringing up the nucleotides now. DNA polymerase, the work of DNA polymerase, if you look, where is this thing? DNA polymerase, what is it doing? It is showing over here. I did not write over here, I guess. Okay, I did not put here in the slides, but you can look into the book. So DNA polymerase is an enzyme which is actually bringing, it's usually joining into for the polymerase reaction. Okay. It is actually putting up, adding the nucleotides. One by one nucleotides are coming, and it is actually joining together to form the newly strand. So if you look into the total template, what you can say, points over the DNA replication. First of all, it is the semi-conservative because the parent one strand, newly strand. That polarity is very important because 5' to 3' direction it is moving. DNA polymerase is the enzyme that is actually adding up the nucleotides to form this leading strand. Base pairing is very important because whatever base pairs are there on the parents, based on that, the nucleotides are coming up and then being added into the new strand that is being formed. Origin of replication I discussed, where it is starting point. Replication fork, where it is forming a fork-like structure for the replication. Leading strands and the lagging Okazaki fragments, and Okazaki fragments are finally joined in the with the help of ligase enzyme. Clear, class?
Ma'am, one.
Yeah.
Ma'am, can you show the 24th slide? Ma'am,
24th.
there I was having 24th
Yeah.
slide.
This one?
Ma'am, if in the question these blue lines are given and we are told to draw the leading strand and lagging strand, means we have to draw from this means how to draw that means I mean these two both are same, no? Like we have to draw from the 3-1. The leading strand will be from 3-.
Leading strand will from the tree. I did not get your point. I mean, don't have to. I mean, if you are asked to do the DNA replication, then you have a schematic diagram. You can show this thing. Okay. If I ask
No,
you to
ma'am.
write.
Actually, his question is that the two blue lines are given to us.
Hmm.
Then how will we identify that which one will be the leading and which one will be lagging for the drawing purpose? How we can identify?
Then how will you identify? You tell me now. Just now I told you that thing.
Yes, ma'am, we can.
You have to look into the.
Dna polymerase can only add from 5' to 3'. So we'll think of 3' to 5'. That
Exactly. You have to look into the polarity. The 3' prime and 5' prime, that is very important. The strand, which is actually this 3' to 5' end, that will be our first template on which the leading strand is being formed. Leading strand, because the DNA polymerase works in the 5' to 3' direction. So this will start with this direction, 5' to 3'. So the template will be 3' to 5'. Clear? Similarly, again, DNA polymerase is bringing up here nucleotides from 5' to 3' direction. So in this case, the lagging strand is formed in the 5' to 3' direction in the other template, which is from the 5' to 3' prime.
Okay, ma'am, understood.
Understood? As I said, there is a very nice tutorial I will share in the MS Team, also in the YouTube, very nicely explained, because you know what is actually we cannot show the animation over here. I can show in the symmetric diagram, 2D diagram, but there is a very nice video where they have shown the 3-dimensional how this DNA replication. There are very nice animations. So I think you just look into that, you will be understanding what is actually happening. Okay, but try to remember enzymes name. The enzymes names end with ASE, always I said. So DNA polymerase, DNA ligase, two enzymes I discussed, but then there are other enzymes like DNA gyrase and all. You can just have a look into the books if you want. Okay. So DNA replication, if you have understood now, if it will be easy for you to understand the transcription, how DNA forms the RNA. Okay. Where will we go for this? Okay. Yeah, one few things that is very needed, that is important in DNA replication, is that while the DNA is formed, there is a phenomenon, there is a method known as DNA repair, which is actually it's very important because there might be some mistakes in the machinery. So for checking these mistakes, that the DNA replication, DNA repair is being done. So sometimes what is happening is that radiation and toxic chemicals damage this DNA, or there is some mistakes in copying the damaged DNA. So DNA repair machinery. So DNA has this machinery which is actually repairing this mistake. So usually this happens properly, but if sometimes it doesn't work properly, then it leads to the different diseases and all. Okay. Usually DNA has this machinery to repair these things if there is some mistakes. So this checking for this mistake is known as DNA proofreading. So DNA is very smart. It has this machinery to proofread itself. So sometimes mistakes can be incorporated by DNA polymerase. Okay, because it is adding up how many? 1000 bases per second. Can you imagine? In just 1 second, in fraction of a second, when I'm discussing so many things, it is adding up 1000 bases. Right now, what is happening inside your body, that cells are dividing, DNA is dividing, and in just 1 second, 1000 bases are being added. So it might happen that it may end up in ending some wrong nucleotide as well. Okay, so what is DNA is having? DNA is having this proofreading mechanism with the help of same enzyme, the DNA polymerase. So DNA polymerase is a very, very vital enzyme. This is actually going for the DNA proofreading, what is actually happening. This is looking for the mistakes if it is done. Okay, if something is mistaked by mistake, any wrong nucleotide is coming up, it's actually checking that and changing that part. Okay, DNA polymerase. One future mutation is a phenomenon where sometimes it happens that an error might happen. Okay, so if the proofreading mechanism doesn't work or it fails, so this error that like A के साथ T pair होना था, but A के साथ अगर suppose G pair हो गया and somehow G has come into that part and then it is permanently set up there. So because the DNA polymerase does not work over here, it fails, then this error becomes a mutation. There is a permanent change in the DNA sequence. And then what will happen? Because we'll later look out, because DNA is passing the information to RNA and then RNA is passing the information to proteins. Suppose there is just one change in the DNA. So the RNA will also change, and this RNA change, then the protein will also change. And once it becomes, the protein changes, means that is going to alter your total machinery. So that is why you end up with several different diseases and all that. So mutations is actually the source because of which so much variations are also there in this universe, in this world. Okay, so what is happening? Agar everything is going properly, then there is no change at all. So sometimes it is also needed, that is the need of the hour, that some changes are happening because of some errors, some mistakes are incorporated. That's why new things have been found. I hope everyone has watched the movie. Which one? Which one I'm talking about? The Hollywood movie about the mutations.
Resident Evil.
Sorry?
Resident Evil.
Resident Evil. Okay. Now the series one, the mutants.
X-Men.
X-Men. Okay, very good. X-Men देखा ही है ना? X-Men, you have seen
Yes,
how
ma'am.
mutations are being created. Oh, that is very high-end movie drama, but that can be possible. Yes, that is possible. That is a scientific fiction, and that is quite based on these things, the mutations, because they are altering the genetic instructions of the DNA, and which actually sometimes it is harmful, sometimes it is not harmful also. So sometimes the body takes it like that, and it's okay. I mean, something like that again depends on that kind of thing that has been incorporated. When we are looking later on, when we are discussing about proteins, how proteins are being found, you can see that different there are codes we'll discuss. I will so amino acid, the 20 amino acids, let's say lysine, can be formed from a particular set of DNA, a set of DNA codes. So if the same code is being replaced, let's say, how can I explain this thing? Like this, what I can say. I have, let me show you this in the camera. So you can see me. So suppose this pair is actually forming this particular protein, this amino acid, and then this is another one which is kind of similar to this one, is also actually forming this amino acid. So if I am replacing this one with this bit, there might be some differences between these two, but again they are coding for the same protein or the same amino acid, then it is fine. It will be not harmful to you. The body will take it as, and it is okay. But if suppose it's small change is happening over here, which is actually coding for a different amino acid, which is actually changing the entire scenario of your body thing, then it will be harmful for your body. And this one very good example of this is the sickle cell anemia. You have heard about this thing in disease, sickle cell anemia, where our red blood cell, if you look, they are actually in spherical, in a round shape manner. But what happens in sickle cell anemia, just one nucleotide is being replaced over here, one base, and then it becomes kind of sickle sickle जिसमें धान काटते हैं and all that. It looks like that sickle cell, and that creates so much a problem in that particular human being. So mutations, that is what about mutations. A small thing can create so much of problems also, but if it is quite similar to it, the body actually takes it, so it might not be harmful also. You cannot say that mutation is always good or always bad. Sometimes it can be harmful, sometimes it might not be. But definitely mutations are actually the reason behind we have so much of variety, so much of changes, so much of evolution is happening, because change is taking place, and that change, it is not like that एक change हो गया तो अचानक से पूरा change हो जाएगा. It takes thousands and thousands of years to get incorporated, and gradually there is evolution is happening. So if you look into the evolution, how humans have appeared, if you go back to the phylogenetic tree, phylogeny, from the small prokaryotes, the eukaryotes have developed, and then you look into the bigger animals, then from the aquatic animals, the fishes, to the amphibia, then the land animals, then the birds, then the reptiles, and then finally the mammals and all that. So this has taken thousands and thousands of millions of years, and the small changes has been incorporated. Gradually the body takes it, it accepts it, and then variations occur. Okay, so
Ma'am, one thing,
Yeah,
like
yeah.
the coloring of the cornea, that is also kind of mutation only, no? Like
So many
someone
things,
is
any changes are happening, you know, that things are really because of the thing, that original things, some will be something different than, just like, you know, if you are an artist, you can say you are taking a picture, that's an original picture. When you draw, you will be drawing some changes you will be using. I will draw something, changes will be happening. But again, the same thing, I mean, that becomes modified. When I keep it, gradually keeping it, I will put into the internet, you will put in the internet, then someone will take your picture as the part one, and it will start drawing on that picture. That happens, so that changes will happen. इसमें आपने blue आंख बना दिया था, मैंने green eyes बनाया था, then someone will take my picture as the source, and then it will make, so just like, I'm just giving a blank example, it is not the same thing with DNA, but I'm just trying to correlate with each other. Okay, so that is about mutations. So I'm summarizing DNA replication. Enzymes, DNA ligase is that enzyme that actually seals the breaks or the gaps in the double-stranded DNA. The DNA polymerase is the DNA replication enzymes. It actually assembles the new strand of DNA based on the sequence of the DNA template. So DNA template, whatever the sequences are there, based on that new data strand is being formed. DNA repair mechanism, as I said now, any of the several processes by which enzymes repair the damaged DNA. DNA replication process by which the cell duplicates its DNA before it divides. And DNA sequence, the order of nucleotides, bases in a strand of DNA. Mutation is the permanent change in the DNA sequence. So in the next class, we'll be discussing about transcription, which is actually formation of the RNA from the DNA. So once you have learned DNA replication, transcription is very easy. It won't be a problem for you. So class till now, any doubts? No doubts. Okay, I'm only in the attendance. Someone has again asked something. Raised hands. Who has raised hands? Bala Raju, you want to say something?
Ma'am?
Yes.
Ma'am, the damage of DNA by the radiation can cause cancer.
Yes.
Or...
So see, cancer has several reasons behind it. So mutations are one of those, but you cannot say that because of only mutations cancer is being caused. There are other things also. Cancer is a very, very, vast disease, which is because of many causes, external, internal, anything. So external को हम लोग control कर सकते हैं, बीड़ी cigarette and पान गुड़ाकू, and then those things, so many advertisement is coming up, you can just go for those and you can stop yourself from them. But if something happening from the inside, what you can do, isn't it? You have seen there are very good people who are actually very health conscious, they never smoke, they never take tobacco and all that, but they can get cancer and all, isn't it?
Yes, ma'am.
So that is because something that is internal thing is happening. So people are doing research, and research is still going on in cancer area, trying to understand what is actually happening because of the reason of cancer and the treatment of the cancer. This is more again important. We do not have a yet proper treatment for cancer. We are trying to find out what is the reason behind it. Okay.
Ma'am, I have heard a term called oncogene.
Oncogenes,
The-
okay.
Is the reason
Oncogenes
behind
is related to the
cancer.
cancer. That is very nice that you have learned about something on oncogenes. Anyone want to add something on this? So some genes are cancer causing, the oncogenes they are saying. So that's what they have, that actually they are not actually causing. The sense is that might trigger. Okay. So there is what about the genes is.
Ma'am, normal
Okay,
person can
thank you,
also
ma'am.
have oncogenes, means in the form of proto-oncogenes.
Yeah, we have.